and diversity in shapes and sizes of plant and animal cells. We shall study plant cells such as palisade cells, guard cells, parenchyma, collenchyma, sclerenchyma, xylem, and phloem with their individual components. Palisade cells. For study of palisade cells, focus a freshly prepared or permanent slide of transverse section of a dicot leaf. This slide has to be observed under the high power of a compound microscope. You can see following details. Just beneath the upper epidermis lies the palisade parenchyma. The cells of palisade parenchyma are columnar in shape. The cells are closely packed and are without intercellular spaces. They are loaded with the chloroplast. Thus, this layer carries out important function of photosynthesis. 2. Guard cells. Guard cells are seen around stomata, therefore first of all focus a freshly prepared or permanent slide of stomata of a dicot leaf under the high power of a compound microscope. You can note the following details. The guard cells are kidney-shaped. They are specialized epidermal cells surrounding the stomatal aperture. The outer wall of the guard cells is thin, while the inner wall is thick. The guard cells possess chloroplasts. 3. Parenchyma. In transverse section of any plant material, you can see parenchymatous cells. Focus a freshly prepared or permanent slide of parenchyma under a compound microscope. The characteristics of parenchyma are: parenchyma is a living tissue. This tissue is made up of isodiametric cells which are either spherical or polygonal in shape. The cells are thin-walled. The cells possess prominent nucleus. There are spaces between the parenchymatous cells. Such intercellular spaces are conspicuously seen when you observe parenchyma. Parenchyma is a fundamental tissue of the plants. Its main function is storage of food. 4. Collenchyma. Take a slide of transverse section of dicot plant material. Observe the structure of collenchyma that is seen under a compound microscope. Collenchyma is a living tissue. The cell walls of collenchyma are unevenly thickened, and the thickening is due to the cellulose. The thickening occurs at the corners. Collenchyma is absent in monocots. Collenchyma gives mechanical support and tensile strength to the organs. 5. Sclerenchyma. Focus a freshly prepared or permanent slide of sclerenchyma under a compound microscope and note: sclerenchyma is a dead tissue. The cell walls are evenly thickened and the thickening is due to lignin. The cells of sclerenchyma being hard and strong. Their function is to offer rigidity and mechanical strength to the organ in which it occurs. 6. Xylem. The xylem is a complex tissue made up of 4 types of cells, that is, tracheids, vessels, xylem fibers, and xylem parenchyma. Tracheids. Tracheids are dead, elongated cells with tapering ends. Their walls are highly thickened due to the deposition of lignin. These thickenings in their walls are of different types such as annular, spiral, scalariform, reticulate, and pitted. Vessels. Vessels are made up of elongated tubular cells placed end to end with the intervening walls dissolved. Vessels are dead cells and have a lignified wall with annular, spiral, scalariform, reticulate, and pitted thickenings. Vessels are seen in angiosperms. Xylem fibers. Xylem fibers are sclerenchymatous cells associated with the xylem. They provide mechanical strength to the xylem. Xylem parenchyma. The parenchymatous cells associated with the xylem are called xylem parenchyma. They store food and water. Function of the xylem is transport of water and inorganic sap from roots to the leaves in upward direction. Phloem. The phloem is a complex tissue composed of 4 types of cells, that is, sieve tube, companion cells, phloem parenchyma, and phloem fibers. Sieve tubes. sieve tubes are made up of many elongated cells which are joined end to end. They possess cytoplasm but lack nuclei. Their end walls are perforated like a sieve and hence they are called sieve plates. Owing to the presence of sieve plates, the contents of one cell is in communication with that of the other. The sieve tubes are seen only in angiosperms. Companion cells. Each sieve tube is accompanied by a specialized parenchymatous cell called companion cell. Companion cells are thin-walled with prominent nucleus and dense cytoplasm. Companion cells communicate with the sieve tubes by means of simple pits. Phloem fibers. These are sclerenchymatous cells associated with the phloem. They are the only dead elements in the phloem. Phloem parenchyma. These are the parenchymatous cells found in association with the sieve tubes. They store food materials and help in their conduction. Let us now study animal cells. We shall learn the details of squamous epithelium, all the three types of muscle fibers, and blood cells. 1. Squamous epithelium. Focus a freshly prepared or permanent slide of squamous epithelium under the high power of a microscope. You can observe the following details. The squamous epithelium is composed of thin and flat polygonal cells. The cells are placed edge to edge like the tiles of a floor. Hence, it is also called pavement epithelium. Each cell consists of a large centrally placed nucleus. Squamous epithelium is present in peritoneum of coelom and also in the endothelial lining of blood vessels. Squamous epithelium helps in filtration and diffusion of substances. 2. Muscle fibers. Muscle fibers are of three distinct types. They are striated muscle fibers, smooth muscle fibers, and cardiac muscle fibers. A. Striated muscle. You can observe structure of striated muscle fibers under the high power of a compound microscope. Use a permanent slide or freshly prepared slide for this purpose. The striated muscle fiber cells are elongated, cylindrical, and non-tapering. The cells are covered by a delicate membrane called sarcolemma. The cells show alternate transverse light and dark bands which give a striated appearance to the tissue. Each cell shows many elongated nuclei which are placed peripherally. Function of striated muscles is to carry out the voluntary movements of the body such as walking, running, moving limbs, etc. B. Smooth muscles. Observe a permanent slide of smooth muscle under a compound microscope and note. Smooth muscle fiber is composed of fusiform cells tapering at both the ends. Each cell shows a centrally situated oval nucleus. The nucleus is surrounded by cytoplasm called sarcoplasm. The sarcoplasm is surrounded by a membrane called sarcolemma. Function. The smooth muscles bring about involuntary movements which are not under our control or will. The peristaltic movement of stomach and intestine, movement of diaphragm, rib cage, etc. are carried out by smooth muscles. Cardiac muscles. When you observe a permanent slide of cardiac muscle in longitudinal section under a compound microscope, you can see the following details. Cardiac muscle is composed of short cylindrical branched cells. These muscle fibers are short, branched with ill-defined sarcolemma. They are uninucleated and joined together by neighboring cardiac muscle fibers. At the union of fibers, there are special zigzag junctions called intercalated discs. The cells also show faint transverse striations due to alternate light and dark bands. Function. The cardiac muscles bring about the rhythmic contractions of the heart. 3. Mammalian blood smear. Focus a freshly prepared or permanent slide of human blood smear under the high power of a compound microscope and observe the following details. The smear shows three types of blood corpuscles, that is, erythrocytes, leukocytes, and blood platelets, thrombocytes. Erythrocytes or red blood corpuscles are somewhat oval and biconcave. They are without nucleus and contain a respiratory pigment called hemoglobin. Leukocytes or white blood corpuscles are of two types depending upon presence or absence of granular cytoplasm. If cytoplasm is granular, they are called granulocytes, whereas the leukocytes without granular cytoplasm are called agranulocytes. Granulocytes are of three types: neutrophils, eosinophil, and basophils. This distinction is due to their staining properties. Agranulocytes are of two types: lymphocytes and monocytes. Each leukocyte has distinctly different nucleus. Blood platelets or thrombocytes are minute disc-shaped cell fragments. They have no nucleus. They play a vital role in the clotting of blood. RBCs or erythrocytes carry respiratory gases. WBCs or leukocytes produce antibodies and are phagocytic. Hence, they protect the body from infections. Blood platelets or thrombocytes are concerned with blood clotting reactions.